Assalamualaikum and very good morning everyone. So welcome to our second Professor lectures for Biology, Pusat Asasi UiTM Campus Dengkil. And my name is Siti Nurfana Muhammad Idris and it's my pleasure to be your moderator for today. And it's an honor to have our guest professor with us today. And we have Professor Dr. Muhammad Faiz Foong bin Abdullah from Faculty of Applied Sciences, UiTM, from UiTM Shah Alam. And we also have in this session our own Dr. Hanizah Siza Abdul Khalil, okay, our Head of Biology Department, Sir Zaidi Yaakob, our Timbalan Pengarah, okay, and lecturers and all students. Now, before we begin our lecture today, to bless our lecture session today, I would like to invite Wan Muhammad Adif to recite the doa. So, silakan Wan Muhammad Adif untuk bacaan doa.
Al-Fatihah. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين بسم الله الرحمن الرحيم الحمد لله رب العالمين والصلاة والسلام على أشرف الأنبياء والمرسلين وعلى آله وصحبه أجمعين اللهم افتح علينا حكمتك وانشر علينا من خزائن رحمتك يا أرحم الراحمين ربنا زدنا علما وارزقنا فهما واجعلنا من الصالحين وصلى الله على سيدنا محمد وعلى آله وصحبه وسلم والحمد لله رب العالمين.
Amin ya Rabbal Alamin. Thank you, Wan Muhammad Adif, for the dua. Alright, on behalf of Pusat Asasi UiTM, we would like to extend a very warm welcome to Professor Faiz, who's going to share his knowledge and expertise with us today. Professor Faiz is a notable academician and he is no stranger to Pusat Asasi as he's giving lectures to Asasi students in the previous years. Now, I believe you all are curious about Professor Faiz, so let me share a brief introduction about Professor Faiz. Professor Faiz is a lecturer at the Faculty of Applied Sciences in the UiTM Shah Alam campus. He graduated with a degree in microbiology from University Malaya, and he obtained his PhD in biochemistry from the University of Oxford. His main area of research is genetic engineering and cellular biology of the model organism Saccharomyces cerevisiae and environmental microbiology. Professor Faiz also held many top positions in various professional organizations, especially related to biorisk and also biosafety. Now, for our lecture today, we're going to discuss chapter 4, starting from subtopic 4.5, cofactor, until subtopic 4.9, enzyme technology. Now, to all students, if you have any question for Professor Faiz, you can write your question in the chat box. And without further ado, I would like to invite Professor Faiz to give his lecture. Okay, Prof.
Okay, thank you very much, Fahana. And Assalamualaikum, everyone. So it's good to see so many young, eager faces here. All right, I think the first thing we do is we should just sit back and relax. Okay, so be comfortable when you are attempting to gain some knowledge. Okay, and we will share what we know. Okay, and learn from each other. Okay, so my name is Faiz. You can call me Dr. Faiz. Okay, and I am a lecturer in the Faculty of Applied Science in UiTM Shah Alam. So I gather that you are all Asasi student who are taking biology. So ni kira semua nak jadi doktor dah ni. Is that true? Ada siapa yang tak nak jadi doktor tak? Okay, that's probably not a good question to ask, isn't it? Okay, but do bear in mind that there are other options. Okay, so biology is a very interesting and a very big field. Okay, and in fact, this century is going to be the century of biology. So biotechnology, and in fact, there's a lot of technology we call new emerging technologies. So these are going to be taking over the world by storm. Okay, so we have seen a few examples of this. Okay, when the COVID pandemic hit us, okay, if that happened 10 years ago, we will be really in serious trouble. Okay? 10 years ago, we probably don't have the ability to detect COVID infections. We probably don't have the type of sophisticated vaccine technology to allow us to produce vaccine in such a quick time. So this is all happening very, very fast. In fact, it's happening so fast that a lot of time we lost our grasp on the technology and how powerful is it. Okay, so my involvement is for the past few years has been a lot on biosafety and biorisk. That's to evaluate whether such technology can be dangerous. So it's also quite interesting. Okay, but I digress. Okay, so let's move on. Okay, before I start, is there anything you want to ask me? Tak ada. Okay. Again, concentrate on the knowledge that you will get. So don't worry too much about passing exam. Okay, there's something that I think most of you are bright, chosen youngsters. So passing an exam is second nature to you. Okay, so don't worry too much. Don't feel too stressed. Okay, allow me to share my strikes. Okay. Okay, can I verify that you can see my slide?
Yes.
Okay,
Yes,
I'm going to put it to a slideshow. This can be a bit tricky. Okay, so can you see my slides?
Yes, Prof.
Okay, sorry, my laptop is set in such a way that when I put on slideshow, I can't see anything else except the slide. So if there's something that you need my attention, you have to just put on your mic and ask a question or whatever. Okay.
Okay, Prof.
All right. Okay. So I believe that in the past lecture you have covered some of these topics. Okay. So we are going to talk about the topics of enzymes. Okay, which is a class of very interesting molecules. Okay. In fact, they are the driving force behind biotechnologies. Without enzyme, we would not have biotechnology and a lot of wonderful things. Okay, so I believe that you have gone through topic 4.1, 2, 3, and 4. Okay, and we will start. We'll begin with topic 4.5 today. Okay, is that correct? Okay.
Yes.
Yeah. Okay. All right. Thank you. Okay, I'll just move very, very quickly. Okay. I'll just flip to the early slide so that you can sort of a quick recap. Okay, so enzymes are catalysts that lower the energy barrier. Okay, they are very specific to the substrate. Okay, it's like a lock and key fit. Okay, and the attachment of the substrate to the enzyme changes the shape of the enzymes and allow it to be active. Okay, we'll talk more about that later. Factors that affect enzyme activities. Okay, so for example, enzyme concentration, substrate concentration, okay, temperature. Okay, and a few equations that you should be able to familiarize yourself with. Okay, how do we measure the velocity? How do we measure the Vmax and the Km? Okay, these are basic enzymes mechanisms. Okay, so all right, and it might come this maintain constant, which is to describe the characteristic of the enzyme. Okay, so every enzyme will have its own Km and Vmax and so on. Okay, and we can use that to sort of evaluate, okay, what is the enzyme doing well, or what is it good for. Okay, all right, and so on. Okay, we're just going to do a very quick recap. Okay, so remember, enzymes are catalysts. They are proteins. Okay, so that's an important point to remember. The enzymes are proteins. Okay, and they are catalysts, meaning that they do not get used up in the reaction. Okay, so what do catalysts do? They catalyze reaction. Okay, so catalysts are components of a reaction that allow the reaction to happen faster and easier. Okay, without being used up in the process. Okay, and the use of enzyme as a catalyst, okay, is best exemplified by this by comparing these two processes. Okay, so on your left, okay, you can see the industrial process for producing ammonia. Okay, so in this process, that we call it the Haber process, nitrogen and hydrogen are combined into the ammonia molecule. Okay, and this happens to be an important process. Okay, anybody knows what is the main use of ammonia? General knowledge. Besides smelling bad, what do ammonia do? One, two, three. Okay.
For fertilizer.
As a fertilizer, thank you. Okay. So ammonia is the main component of fertilizer. So without this process, okay, we may not be able to produce enough fertilizer to grow all the food that we need. Okay, and this process happens in the industry. Okay, so but it requires very extreme condition. So you need to heat the nitrogen and the hydrogen and the iron catalyst, okay, up to about 450 degrees Celsius under a very high atmospheric pressure. So we are talking about 200 times the normal atmospheric pressure. Okay, you put a normal human inside here, you'll be crushed to the size of an egg. Okay, so in our industrial process, okay, this process can happen, right, under very extreme condition. However, okay, biology has found a way to make it easier. Okay, some bacteria, okay, we call them nitrogen fixing microbe, and they like to live in the roots of legumes, kacang. Yeah. So there's a legume plant. You pull one out, you can see that at the roots there are some tiny little knobs, and inside those nodules, there is a bacteria. Okay, and these bacteria are able to do the same process. They can carry out the same process, taking nitrogen and combining it with hydrogen and produce ammonia. Okay, by using an enzyme called nitrogenase. Okay, and all this happens at very mild condition. It happens at normal temperature. In the soil is probably about 28 degrees Celsius and under normal atmospheric pressure. Okay, so this is the power of enzyme. It allows us to do things, okay, in a very much more reduced way. Okay, just one moment while I change my cursor. I think I will take a pen today. All right, okay. Hope too small. Point option. What color do you all like today? Red, okay, boleh. All right. Okay, so the same process, okay, can happen, okay, by using an enzyme called nitrogenase, okay, at a very mild temperature and atmosphere. Okay. So this is what we mean by that enzymes are catalysts that lowers the required energy of activation. Okay. Without enzyme, you need a very high energy of activation, high temperature, high pressure, and so on. With the use of enzyme, okay, we can reduce the energy that's required for the reaction, and it happens just at room temperature. Okay, so this is one example of enzyme. Okay, the other thing that enzyme impact on us recently, okay, if anybody has the chance to do a COVID-19 PCR test, anybody was required to do a swab test, were you required to do a swab test before you can return to Dengkil? Hey, wait a minute, are you in Dengkil now or at home?
Both.
Some of us at Dengkil.
Oh, okay, so not everybody is at home. Okay, so anybody has the chance of doing a swab test? Nobody. Okay, you're all lucky people. Okay, so when you do a swab test to test for whether you have been infected by COVID-19, okay, so the lab would take a sample from your nose, okay, and do what you call it, a PCR test. Okay, and the PCR test is made possible by the use of two enzymes. Okay? The first one is the reverse transcriptase, okay, and the second one is a DNA polymerase. Okay, these are the things that can work hard to diagnose whether you have COVID-19 infection. Okay, now both of them do the same thing. Okay, so both the reverse transcriptase and DNA polymerase, they perform the same function. They make new DNA. Okay, as you can see in the picture. Okay, the difference is that RNA polymerase is specific to RNA, and the virus, the COVID-19 virus, has an RNA genome. It doesn't carry DNA, unlike most organisms. Okay, the COVID-19 virus carries an RNA genome. Okay, so we can only find RNA, virus RNA. We don't find virus DNA in your sample. Okay, so the first step is reverse transcriptase will act on the RNA and produce a DNA. And then DNA polymerase, okay, will pick up the DNA and make more DNA. Okay. So although they do the same function, okay, but the substrate are different and they're very specific. So the reverse transcriptase will not work on DNA, or the DNA polymerase will not work on RNA. Okay. So this is yet another example of enzyme characteristic. They are very specific to their substrate. Okay, and you also learn in the first part that certain factors affect the activity of the enzyme that is under study. Okay. One example is temperature. When you heat an enzyme, okay, you find that the temperature, when you increase the temperature for an enzyme, you find that its activity goes up. Okay, up to a certain point when, okay, the enzymes start to degrade, okay, and then the activity drops. Okay, so now the PCR technology that was used in the previous COVID-19 test was made possible by the discovery of what we call thermostable enzyme. These are enzymes that are very stable to temperature. Okay, so this is how we do PCR. This is the PCR process. It involves heating the enzyme and the DNA, okay, up to 94 degrees Celsius for a minute. And then we lower down the temperature to 55 degrees Celsius for the DNA to anneal back. And then we increase the temperature again to 72 degrees Celsius to allow for the DNA polymerase to work and make more DNA. Okay, this happens at least 35 cycles, 35 times. We keep doing it. Okay, and you will notice that, okay, even though we heat it up to such a high temperature, okay, the enzyme is still remain active. Okay, although the activity dropped to very low at 94, but when you take it back to 72, okay, the enzyme become very active again. Okay, so different enzymes will have different temperature range. Okay, and we have enzymes that are very, very thermostable. There are some enzymes that has an optimum temperature of 105. Okay, boiling water. Okay, but it happily live in there and laugh and do it work at 105 degrees Celsius. And there are enzymes that will work very well at 4 degrees Celsius. Okay, low temperature. Okay, some organisms that live in the Arctic, Antarctic, they need enzyme that can work at low temperature in order for them to survive in such environment. Okay, so they're wonderful things, enzyme. Okay, you find a large range of them, and with all different types of characteristics. Okay, so that with that we are done with a bit of recap of the previous lecture. So I'll move on. Before that, any question? Anything you'd like to ask? Again, I'm sorry, because my screen, I can't see the Google Meet. So if you raise your hand, I will not be able to see you. Okay, we're all right there? Okay. A bit quiet on your side. Anybody want to try ask a question?
No, Doctor.
Oh, yeah, okay. You're all right. I keep hearing the same voice. Okay. Let me see whether I can... okay, just one moment. I'm not sure why is it in this way. I cannot see the... okay, I think I better not. Nope, I shouldn't do this. Okay.
Prof?
All right, okay.
Prof, can you hear me?
Yeah, I can hear you.
I'm Zaidi. When you mentioned just now the process of the technique of PCR test just now,
Okay.
we have to use the enzyme DNA polymerase. Do we have to use specific DNA polymerase? Do we have to use the specific DNA polymerase from specific organisms?
There are a few. The one that we normally use is called a tag polymerase, simply because it's one of the best. It's robust. Okay, but there are other organisms that also produce DNA polymerase. For the PCR test, the most important thing is that it's stable to temperature, because the test is conducted as the graph shows us now. You have to heat it to 94 degrees Celsius. Okay.
Right. Thank you, Prof.
All right, thank you. Okay, I'm trying to adjust my screen, but I still cannot. Sorry about that. It's just a glitch that I didn't expect. And I'm unable to. Okay, maybe I will just open another. I will just duplicate my screen. Just one moment. Okay, I will just duplicate my screen, and I will join in again. Okay, but then I need to off my video. Okay, and then I can share this. Okay. Okay, no, still doesn't work. Okay. Sorry, just a moment. I just get back into it. Okay, I guess we just have to continue like this. So you need my attention, just shout in the phone. Okay, you know the mic. Can you still hear me?
Yes.
Okay. Right. Okay. Sorry about that. Okay. Now let's continue with the topics. Okay. So we're going to talk about cofactors. Now some enzymes require cofactors. Okay. So a cofactor is another molecule that is required in the reaction. Okay, but again, it's not used up in the reaction. Okay, and it helps the enzyme in doing its work. Okay, so an apo enzyme is an inactive enzyme. Okay? That is where it's on its own. Okay, and it requires a cofactor to be functional. In fact, most of the enzymes that in our body they require a cofactor to work. Okay, so cofactor also allow the enzyme to be controlled in a certain way. Okay, we'll see more about that later on. Okay, so when you provide a cofactor or a coenzyme to the enzyme, okay, then you form a holoenzyme. Okay, so the enzyme with the cofactor together becomes a holoenzyme, and in this stage the enzyme becomes active. Okay, so from an inactive stage, okay, when you add the coenzyme, it becomes an active enzyme. Okay, and then it can start to pick up the substrate. Okay, and continue with the reaction. Okay, so this is actually a form of regulation of how you control enzyme. Okay, so we have to bear in mind that enzymes are very powerful molecules. Okay, so for example, a digestive enzyme like trypsin, it digests protein. So a trypsin is an enzyme that will cut and break down your protein into amino acids. So if it work on protein, it can work on your protein, and your cells are made up of protein. Okay, so you simply cannot have the trypsin enzyme running wild in your body. Okay, it will quickly reduce you to a mesh of a watery substance. Okay, so we need to control the activity of enzymes. Okay, and one of the way is to the requirement for a core enzyme, so that it's only active when you want it to be active. If you don't want it to be active, take away the core enzyme, and it becomes inactive. Okay? So it's a sort of like a switch on, off switch. Okay. Okay. So there are basically two types of cofactors. Okay, organic or inorganic. Okay, and remember, a cofactor is a non-protein molecule or ion needed for the proper functioning of an enzyme. In order for the enzyme to be working properly, you may need to provide a coenzyme or a cofactor. Okay, so organic cofactors, okay, are generally known as coenzymes. Okay, and they do important function, okay, that helps the enzyme to transfer certain chemical groups, atoms, or electrons. Okay, so you can see in this example given, okay, NAD, okay, nicotinamide adenine dinucleotide, okay, is a ubiquitous coenzyme in your body. It's involved in a lot of reaction. Okay, so the enzyme malate dehydrogenase catalyze this process. It changes malate to oxaloacetate by removing 2 hydrogen atoms. Okay, now when you remove these 2 hydrogen, okay, it has to go somewhere. You don't simply want active hydrogen atoms running around in your body. Okay, it can cause a lot of trouble. So the enzyme knows that. So when it removes the hydrogen atom, okay, it has to go somewhere and is picked up by the core enzyme NAD. Okay, so NAD picks up these 2 hydrogen and becomes NADH plus a hydrogen ion. Okay, so then it will go somewhere. Okay, so the NADH2 with 2 hydrogen you'll probably go to another reaction where the hydrogen is donated to that reaction. Okay, so this is how biochemical reactions are precisely controlled in your body, okay, by enzymes. Okay, so they know that when they do this, this will happen. How do I take care of this problem? Okay, how do I remove this hydrogen? Okay, I use NAD to take it to somewhere else which require hydrogen, and that can be used in that other reaction. Okay, so that's basically what a coenzyme does. Okay, so there are many different types of coenzyme in your body. Okay, NAD is a very common, very major one. Okay, and its partner NADP. Okay, with the added phosphate group. Okay, ATP is a common coenzyme. Okay, adenosine triphosphate, the energy molecule, and also other molecules like coenzyme A. Okay, as you move on to your future chapters on respiration and photosynthesis, I think there's chapter 7 and chapter 8. Okay, you'll see a lot of this in action. Okay, how they are used in the respiration process and how they are used in the photosynthesis process. Okay, so besides organic coenzyme, okay, we also have inorganic cofactors, and these are usually metal ions. Okay, like calcium, okay, zinc, magnesium, and so on. Again, they do not form part of the reaction, but they are required. Okay, so enzymes require them to be there, okay, in order for them to work well, okay, in catalyzing the reactions. All right, and you will actually find that a lot of the vitamin and minerals that you are asked to take as supplement. Okay, I'm sure some of your parents has provided you with vitamin C, okay, and a lot of things like that. Okay, anak makan banyak-banyak sikit, okay, jaga kesihatan, okay, masa ketengkir ni, okay, jangan makan KFC, McD saja, okay. They are actually cofactors and enzyme, okay, that your enzymes require to work. Okay, so one example is coenzyme Q. Anybody uses this? Siapa makan tak coenzyme Q ni? Coenzyme Q10. Used to be very popular, and I think masih popular juga. Anybody?
No.
No. Okay. All right. Okay. So then you people are good at keeping yourself healthy. Okay. Tak ada siapa nak jual yang buat MLM ke? Thank you ni ke? Jual coenzyme Q10 ke? Once upon a time, it's very popular. Everybody is selling coenzyme Q10. Okay. So this is a coenzyme. Okay. So it's an important coenzyme in the respiration pathway. This is what happened when you breathe. Okay. When you breathe, you rasa senang saja, sedut udara saja. But this is actually what happened in a cell, in your mitochondria when you breathe. A very complicated series of chemical reaction. And the red dot here is coenzyme Q10. Okay, so it functions to transfer electrons from one enzyme to the other to another enzyme. Okay, and a lot of people will tell you that, okay, as coenzyme Q10 is important in the respiration pathway. Okay, so it will allow you to have more energy. You take a lot of coenzyme Q. Okay, it's up to you whether you want to trust them. There is no really strong medical evidence. Okay, but maybe a small amount that would not be harmful. Okay, so this is one example of coenzyme. Okay, and vitamin C is a coenzyme that is required for several enzymes that are involved in the biosynthesis of the protein collagen. Okay, so yang ni budak perempuan suka lah. Okay? The collagen is the protein that makes your face look tight and nice. Okay, hilang kedut-kedut semua. Okay, anybody taking collagen supplement here? Ada?
No.
Tak ada juga. Okay. Alright. Okay. I think you're very sensible young people. Okay. Alright. So you don't need to take collagen. Okay. You take it, you just get digested in your stomach by another enzyme. Okay. So you end up with just amino acid. But you can take vitamin C. Okay, because it's a coenzyme for the enzymes that make collagen. Okay. So if the enzymes are active, then you get more collagen. Okay. So nanti di chain muka tu cantik saja. Okay, tak macam saya dah kedut dah. Okay, all right. Okay, so that is coenzymes and cofactors that are required for enzyme activity, and we are going to see how they are used, okay, to control enzyme. Okay, so in the next topic, we are going to talk about enzyme inhibition. We are supposed to take a break at 9:40, so somebody remind me. Okay, oh sorry, 10:40. All right, 10 minutes more. Okay. All right, so inhibitors are chemicals that selectively inhibit the action of specific enzymes. Okay, so if coenzymes, in a sense, activate the enzyme, or cofactors, in a sense, activates the enzyme, inhibitors are chemicals that now inhibit the enzyme. So these are the off switch. Okay, if the coenzymes and cofactor, you can think of them as on switch. They turn the enzyme on. Okay, but now we want to turn the enzyme off. All right. And again, there are two classes. So one class are reversible inhibitors. Okay, so these are chemicals that will form weak bonds, such as hydrogen bonds with the enzyme. So they come to your enzyme and they bind to your enzyme in such a way, okay, using weak hydrogen bonds that can be broken easily. That's why they're called reversible. Okay, so they can bind, but you can also break the bonding, okay, and remove them. Okay, so when the inhibitor bind to the enzyme, okay, it changes the shape of the enzyme. That's usually what happens. Or it occupies active site of the enzyme, okay, and block the substrate from coming. Okay, and the enzyme becomes inhibited, become inactive. Okay, when you remove the inhibitors, okay, as I mentioned, the bonds are weak. There are ways to remove the inhibitors. Then the enzyme revert back to its normal active functional structure. Okay, so that is what reversible inhibitors do. It allows you to switch off the enzyme when you want to, and then switch them on again, okay, when it's time to switch them on. Okay, and among the reversible inhibitors, there are two types: competitive and non-competitive inhibitors. Okay, we'll take a look at this in a short while. Okay, on the other hand, there are also irreversible inhibitors. So these are chemicals that bind very tightly, okay, and permanently to enzyme. Okay, they form covalent bond. So once they are bonded to the enzyme, okay, it's very difficult to remove them from the enzyme. Okay, and in this case, then your enzyme is more or less, okay, destroyed. Okay, they are permanently deactivated. Okay, so such inhibitors seldom occurs naturally in our body. Okay, they are usually chemicals that are introduced from the external environment. Okay, inhibitors are chemicals that is produced by your own body, okay, or from the external, and you use them to control enzyme. But irreversible inhibitors are just chemicals that you really don't want to be in your body and messing up your enzyme, because the effect is irreversible destruction of the enzyme. You don't get the enzyme back. You have to make new enzymes. Okay. All right. So these are competitive and non-competitive inhibitors. Okay. So as mentioned, they form weak bonds, which are easily broken, okay, such as hydrogen bonds. Okay, and inhibition only takes place when the inhibitor is attached to the enzyme. Okay, when you remove the inhibitor, then the enzyme becomes active again. Okay, so again, think of it in the sense of a switch, like a light switch. Okay, when you turn it on, the light comes on. I mean, when you turn it off, when you flip it the other way, the light goes off. Okay, so in this case, okay, so this is a normal enzyme in the substrate. Okay, so the substrate can bind to the active site, and the enzyme will happily carry out its duty. Okay, but when you introduce a competitive inhibitor, okay, so it's called competitive because it will compete with the substrate for the active site. Okay. When it's bound to the active site, then it prevents your substrate from binding, okay, and no enzymatic reaction happen because your substrate can no longer bind to the active site. So until the inhibitor is removed, okay, the enzyme is switched off. It cannot carry out its function. Okay. We also have non-competitive inhibition, okay, where the inhibitor binds to another site of the enzyme. It doesn't bind to the active site, okay, but by binding to the other site, okay, it's sometimes called an allosteric site. Okay, it changed the shape of the enzyme. Okay, nampak, okay, jari dia dah berubah dah. Okay, here the finger is up and here the finger is down. Okay, the shape changes. Okay. And due to this change of shape, again, the substrate can no longer bind to the enzyme, and no enzyme activity occurs. Okay, so reversible inhibitors, okay, are usually the competitive inhibitors that are reversible, okay, are usually chemicals that has a molecular shape quite similar to the natural substrate. Okay, so if you have a substrate with a certain molecular shape, okay, you'll find that the reversible inhibitors, okay, will also have the more or less similar shape. In fact, they can be more or less very similar chemical structure, okay? And the enzyme cannot differentiate between them. Okay? So sometimes the enzyme will pick up the substrate, and sometimes the enzyme will pick up the inhibitor. Okay, and because of the similar shape, it can bind to the active site. Okay, and it competes with the normal substrate for binding to the active site of the enzyme. Okay, so this generally slows down the reaction of the whatever the enzyme is trying to do. Okay, so reversible competitive inhibitor seldom switch off everything totally. Okay, because you have to imagine that, okay, the in your cell, okay, binding doesn't occur, okay, permanently. This is reversible. So the inhibitor will bind for a short while and then it will detach. Okay? And then again, you start competing again in the next round, whether the enzyme will pick up a substrate or the inhibitor. Okay, so a certain percentage of the time, the enzyme will pick up the substrate and some reaction can still carry on. Okay? So it slows down the rate of reaction, but it doesn't totally stop the reaction. Okay, it interferes with the active site, so the substrate cannot bind easily. Okay, and because of that, less product is produced. Okay, so because of this nature, because of this competitive nature and dynamic nature, where both the substrate and the inhibitor are actually competing to bind. Okay, if the inhibitor wins, then no reaction. If the substrate wins in the binding race, okay, then there is a reaction and you have a product being produced. Okay, and because of this nature, you can actually overcome reversible inhibition by increasing the substrate concentration. Okay, so you add more substrate, okay, then the chances of the enzyme binding with the substrate becomes higher. Okay, so imagine there are 10 substrate for every inhibitor. Okay, then the probability of the enzyme binding to your substrate is much higher than it will bind to the inhibitor, and you will get more proper binding and more reaction and more product being come up. Okay. Okay, so these are examples of enzymes, okay, that uses competitive inhibitors. Okay, so one example is succinate dehydrogenase. Okay, so the enzyme usual substrate, okay, is a succinate. As the name suggests, you can more or less guess what the enzyme do by looking at its name, succinate dehydrogenase. Okay, so most enzyme, when they are, the name has a ASE at the end there. When you see ACE, then you know it's most likely to be an enzyme. Okay, so succinase dehydrogenase, you know, from here you can more or less guess that the substrate is succinate. And what does it do? It removes a hydrogen. Okay, so this is the normal reaction. Okay, the enzyme will pick up a succinate, okay, fix it into the active site, okay, and after removing a hydrogen, it produces a fumarate. Okay, so okay. Now malonate is another molecule, okay, with a structure quite similar to succinate. You can compare it. Okay, both the end are similar. Okay, the only difference is that while succinate has 2 methyl group, malonate has only 1 methyl group. But that is enough to allow for malonate to also bind to the active site of the enzyme. Okay, because the binding occurs via the carboxylic group at both ends. Okay, so that 1 liter extra methyl did not interfere much in the binding. Okay, so malonate can also bind to the same enzyme. Okay, but because it's not the correct substrate, okay, no reaction is catalyzed and no product is formed. So in this case, if you increase the concentration of succinate in relation to malonate, okay, then you have more, you push the reaction towards A. Okay, more of this will happen, and you do get more product. Okay, I think we'll stop here for a little while. Okay, is there any question? We'll take a 10-minute break before we continue to the next section. So is there any question that I can answer for you?
Doctor,
Yes.
I just want to ask, okay, is that true that in order for the enzyme to become active, there must be cofactor, right?
Most enzymes will require a cofactor, although there are some enzymes that doesn't require cofactors. But I think it's a general statement that okay, most enzymes will work better with the cofactors. There's certainly some enzyme that doesn't require cofactors, like some proteases. Okay, so they can work on their own.
Oh, okay. So in order to the reaction to occur, so there must be substrate for the reaction. But for the enzyme to be active, there must be cofactor, is it?
As I said, most of the time, yes, but not absolutely. Okay? So there will be enzymes that doesn't require cofactor, okay, and they can work, okay, with happily without cofactor, although the rate of reaction may be slower. Okay? One example is a protease. Okay? You do your laundry. Okay? When you look at the detergent powder, sometimes you can see some blue dot. Okay? Perasan tak masa basuh baju tu? Atau you tak basuh baju, ada laundry service. Okay?
Yeah.
So, kalau guna sabun putih tu akan ada, sometimes you have the blue dots kan. Those are actually enzymes. Those are proteases. Okay? And they can work well without cofactors. Okay? So it depends on the enzyme.
Okay, thank you, Doctor.
Okay, if there are no further questions, so can I suggest we take a short break? Okay.
Okay,
All right.
Prof. All right.
We can come back at 10:50. Is that correct?
Yes, Prof.
Okay.
10:50.
All right. I'll try to sort out my PowerPoint. Okay.
Okay, Prof.
Sorry about
All
that.
right. Yeah. Okay, everyone. So we're going to continue at 10:50. Right. So stay tuned. Okay, everyone. So I hope everyone is feeling fresh. So we will continue our lecture today with Professor Faiz. Okay, Prof. Can we continue? Shall we?
Okay, thank you very much. So I think I partially solved the problem by converting to a PDF. So if you will let me know whether is this can you all see it clear enough?
Yes, clear, Prof.
Okay, all right. So I'll just use a PDF instead of a PowerPoint if that's okay with you all.
Yeah.
Okay. All right. Okay, thank you then. Okay, so all right, we stop here just now on competitive inhibitors. Okay, so we move on to non-competitive inhibitors. Okay, is my voice still clear?
Yes,
All right. Okay, thank you.
Clear,
Okay, thank you. All right. Okay, so in contrast to competitive inhibitors, okay, so non-competitive inhibitors, they do not bind at the active site. Okay, they bind to another site known as the allosteric site. Okay, so for that, they do not need to have structural similarities with the substrate. They did not need to look like the substrate. They can be a completely different shape, okay, because they're going to bind to another site. And binding to the allosteric site, okay, so cause the enzyme to change shape. So again, a lot of things that happens to enzyme is something change their shape and they're no longer active, okay. So when the shape change, okay, again, it prevents the substrate from binding and the enzyme no longer function normally, and you have a lower rate or a complete stop of the reaction, okay. So this is in contrast to the competitive inhibitors. Okay. All right, and you can know whether an inhibitor is competitive or not competitive, okay, by looking at the rate of reaction of the what you call it, of the enzyme activity. So I just try to make it a bit bigger. Okay. Sorry for this technical. I'm not sure why my PowerPoint chose to misbehave this morning. Okay. So a normal enzyme reaction, you look at the green color curve. Okay, when it's not inhibited by any of the reversible or non-reversible inhibitors. Okay, so follow the normal Michaelis-Menten path. Okay, so it will the rate of a reaction will increase as you increase the substrate. Okay, more substrate, the faster it binds, the faster it works. Okay, until a certain point when it becomes saturated. Okay, there's a Vmax. That is the maximum velocity, the maximum rate of reaction that the enzyme can achieve. Okay, so at this point, no matter how much substrate you add, it will not increase the rate of reaction any further because the enzyme is simply saturated. There's no more enzyme to bind, too much substrate and no more enzyme. Okay, when you add a competitive inhibitor, okay, you'll find that when you have lower substrate concentration, the competitor will compete with your substrate, okay, to bind to the active site, and they reduce the rate of reaction. Okay, so at the same substrate concentration, you have a lower rate of reaction. Okay, however, as the substrate concentration increases, okay, that inhibition effect can be overcome. If you have more substrate, then the chances of the enzyme binding a substrate and not an inhibitor becomes higher. Okay, and that helps with the rate of reaction. Okay, and the system can actually reach Vmax. You will give it enough substrate. Okay, somewhere when you hit the saturation point, okay, the reaction, when there's so much substrate that it completely overwhelms the inhibitor, then okay, the effect of the inhibitor becomes negated. Okay, whereas when you have a non-competitive inhibitor, okay, the picture is a bit different. Okay, because it doesn't compete. Okay, so at some point, no matter how you increase the substrate concentration, okay, it will not have any effect, okay, in increasing the rate of reaction. Okay. When all the inhibitors has bind to your enzyme, okay, then that's the reaction. That's the point when you cannot increase the rate of reaction any further. Okay, is that all right? Any question on this part?
No, nothing.
Okay, so remember that competitive inhibition, okay, is always temporary. You can reverse the reaction by removing the inhibitor. The same with non-competitive. Okay, because they do not bind permanently to enzyme. Okay, it's possible to maybe add another chemical and to remove the inhibitor, and then your enzyme will be left alone to perform as normal. Okay, so these are just molecules that compete with your normal substrate or compete with the allosteric site. Okay. Oops, I lost it again. Okay, here we are. Okay, so that is reversible inhibitors. Okay, and we'll now take a look at irreversible inhibitors. So these are inhibitors that bind to enzyme non-reversibly, meaning that once they bind to the enzyme, they do not let go. Okay, and it's very difficult to remove them. Okay, so they bind tightly and permanently to enzymes. Okay, using strong covalent bond. Okay, they do form a chemical bond, and the end result is that the enzyme's function is destroyed. Okay, so the enzyme no longer function. Okay, and this can occur even at low inhibitors concentration. Okay? Because once they destroy your enzyme, okay, so it can no longer rescue the enzyme. So the reaction can no longer be rescued. Okay, it's like a murder, so you cannot bring it back. Okay, so a good example of a irreversible inhibitor is one moment. Okay, is some of this nerve poison. Okay, for example, sarin gas. Okay, this is a very quite popular nerve gas that can be used to kill. In fact, it's used in warfare. Okay, to attack soldiers and so on. If you remember, I'm not sure I should be talking politics now, but if you remember last year, we have a high-profile assassination case in Malaysia. Somebody was murdered. A North Korean was murdered at our airport. Okay? That happens to be a close relative of a ruling party of what we call a nefarious state country. Okay? A country that doesn't follow the usual regulation, and that act of murder was conducted using something like this. Okay, it was a neurotoxin, a nerve toxin that inhibits an enzyme. Okay. And it works very fast. Okay? I think the killing was that it kills that person in less than 2 hours. Okay? So what things like this do? This sort of nerve toxin do. Okay, for example, sarin. They have no odor, no taste. So it's very hard to defend yourself from there. Somebody could be squirting water on you, and you thought, okay, it's just some playful gesture, but it could be sarin or some other toxin that target your nervous system. Okay. What sarin does is binds covalently to the R group, okay, of an enzyme called acetylcholinesterase. Okay, so when it binds to this enzyme, acetylcholinesterase, it changes the shape of the active site, okay, because it's binding to a group, okay, and the function of acetylcholinesterase, okay, is to break down acetylcholine, a neurotransmitter. So when acetylcholinesterase is rendered inactive, okay, by the binding of sarin, okay, so it cannot do its function. It cannot break down acetylcholine, okay, and because of that, acetylcholine will accumulate in the synaptic cleft. Have you all done cell biology in nerve cell?
It's going to be next semester, Prof.
Okay, next semester. Okay, so we are okay. Tapi ada dalam nota ni. Okay, tak apa. Alright. Okay. Okay. Anyhow, it's alright. Okay. So acetylcholine accumulates in the synaptic cleft, where the junction between the two nerve cells. Okay, I'll show you a picture in a short while. Alright. And because of that, the signal is continuously transmitted. Okay. So what happens when you want to move a muscle? Okay, contohnya you nak gerakkan satu jari ke? Okay, your brain sends a signal. Okay, that signal is sent through a nerve cell. Okay, so there's a nerve from your brain that goes down to your spinal cord, and from your spinal cord there's a major ganglion where the nerve cell travel to your hand. Okay, and as it goes along, the signal is passed to the next nerve cell. Okay, so below the next nerve cell that controls your finger get the signal. Okay, it twitch a bit. So you move your finger, okay? The muscle contract, so you move your finger, okay? Just like what I'm doing now, okay? So I can, when my brain say, okay, muscle move. Okay, muscle move. I bend my finger. Okay. So now this again is a reaction that has to be controlled, because after I've bent my finger, I want to be able to move my finger again. Okay. Jangan signal tu dah sampai, tak boleh patah balik pula. Okay. So for the rest of my life, my finger macam ni saja. You don't want that to happen. Okay. So that signaling is done by a molecule, a chemical called acetylcholine. Okay. So once acetylcholine has done its job, okay, I have bent my finger, now I want to straighten this. Okay, acetylcholinesterase now break down the acetylcholine. Okay, so that release the signal. Okay, now I can straighten my finger. Okay, what happened is acetylcholine is not removed. Okay, then the signal will keep continue. The signal will be passed as though is coming non-stop. Okay, and my finger will be stuck like this. But the muscle doesn't want to react because it is getting the signal to keep contracting. Okay, so this is what sarin does to you. Okay, so it stops the signal from being broken. Okay, and your muscle will continuously contract. Okay? So you can imagine what happened. Your whole body tu, your muscle tu asyik tegang saja. Okay, you can't breathe. Okay, you'll be your whole body cannot relax. It'll be just macam tu. Okay, and that will lead to convulsion, paralysis, and eventually death. Okay. When all your energy is used up to contract your muscle, you will just die. Or you cannot breathe because your lungs muscles are contracting non-stop. Okay, so I added this slide. Okay, so it's probably a bit what you call it, jumping the queue, but doesn't matter. So this is the picture of the synaptic cleft. Okay, you will notice the the synaptic cleft. Okay, yang ini. You can actually highlight. Okay. Oops. Oh, doesn't. Okay, that's what. Okay, so this is a picture of your synaptic cleft. So this is the nerve cell that's sending the signal. Okay, on the bottom side is the nerve cell that's receiving the signal. Okay, this is how your system works. The signal is sent to multiple nerve cells. So the nerve cell that's sending the signal, okay, release acetylcholine, ACH. Okay, and acetylcholine, okay, is passed to the nerve cell that receive the signal. Okay, that's how signal transmission occurs in your nerve cell. All right, and after acetylcholine has done its job, okay, dah pass signal dah, okay, then it's broken down by acetylcholine esterase to choline and acetate, and it's recycled back by the cell for the next round of signaling. Okay, so this process of sending acetylcholine, breaking it down, okay, to release the signal is important. Okay, otherwise, okay, if acetylcholine is not broken down, okay, if I sort of inactivate the enzyme, okay, all right, and this process doesn't happen, then you have acetylcholine here, okay, forever, and your nerve, the signaling will happen forever. Okay, all right. So this is how sarin will kill you by inhibiting an enzyme. Okay. Now the use of acetylcholine as a major neurotransmitter in skeletal muscle is almost universal. Okay. Almost every organism uses this or a variation of this model. Okay, and because of that, okay, many other animals has learned to use acetylcholinesterase inhibitors as a toxin or a poison weapons to kill their prey. Okay, one example is a green mamba snake. So the toxin of the green mamba snake contains a small protein called fasciculin, which is an acetylcholine esterase inhibitor. Okay? So when the green mamba snake bite you, the same thing happen. Okay, the muscle will start twitching non-stop. Okay, it's a painful way to die. Okay, so jangan kacau green mamba snake ni. Nasib baik Malaysia tak ada. Okay, and also some spiders. Okay, spiders have learned to use this sort of chemicals to capture their prey. So when the spider bite the insect, okay, they inject a toxin or venom that contains a lot of this acetylcholine esterase inhibitor. Okay, so that causes the insect to die, and the spider can slowly eat it up. Okay, and we learn from that. Okay, so we were thinking, okay, if spiders can use that, why can't we? Okay, so we also produce a lot of insecticides, okay, pesticide, the thing that you spray nyamuk kat rumah tu, okay, they contain acetylcholine esterase inhibitors, okay, contohnya malathion and chlorpyrifos. Okay, so remember, okay, don't simply play with all these pesticides, okay? Because they can kill insect, but because acetylcholine is universal, they can also kill you at high doses. Okay, don't go running on spraying your friend. Okay, all right. Okay, so this is how enzyme inhibitors can irreversibly, okay, inactivate your enzyme. Okay, the problem here is irreversible. So once the acetylcholinesterase is destroyed, okay, that your body don't have time to make new one. Okay, and the poison act on you before you can make new enzyme. Okay? If it's irreversible, the enzyme is gone. Okay, you need to make new one to replace it. Okay, so that's reversible and irreversible inhibition. Okay, now by using the cofactors to activate and inhibitors to inhibit, okay, so that provides us a way to regulate enzyme activity. Okay, so and again, remember in the cell, okay, the cell is actually a very busy place. Okay, it's a big pasar borong. Okay, with a lot of things happening at the same time. Okay, so sebelah sini orang jual ikan, sebelah sana orang jogging, so on. It's a very big place, very noisy. Okay, very chaotic. Okay, and you need to control all these actions. Okay, you cannot simply let something happen here. Okay, uncontrolled. Okay, or the whole cell will go haywire. Okay, in fact, okay, this is one of the amazing things about biology. Okay, despite all thousands and 10,000s of reaction that goes on in a cell, everything seems to be very precisely organized and controlled. Okay, otherwise we won't be alive. Okay, so that is one of the keajaiban Tuhan. Okay, that it's difficult for us to ignore that things like this cannot simply happen at random. Okay? Thousands of chemical reaction happening in the cell, and they are highly coordinated and highly regulated. Okay, what happens when regulation break down? Let's say in a cell, okay, tak dengar cakap pula, okay, suruh buat macam ni, dia buat macam tu pula. If a cell lost control, what happens to the cell? Anybody has any idea? Bear in mind that we are intelligently created. Okay? Ciptaan manusia ni our biology is in a perfect balance. So if a cell suddenly decides to do something funny, go rogue, what do you think will happen?
Do we have too many products in our body that we haven't used?
Okay, that's part one of the things can happen. You were wasting resource. Okay. Pernah dengar cancer tak? So a cancer is a cell that has lost control. Okay, so usually our cells they are smart. Okay, they don't do things that they don't require to. Benda yang dia sel kita ni sebenarnya bijak tau, dia tak buat benda yang tak faedah. Okay, dia tak macam kita ni lepak kat kedai mamak dan mengetik, tak ada. Okay, dia hanya buat benda yang faedah sahaja. Everything the cell does has a purpose. But if a cell does lose control, okay, sometimes, for example, macam ni some poison comes in, okay, the cell will actually take the decision to die. Cell tu akan bunuh diri, okay, in order to preserve the organism. Okay, it's a process called apoptosis. Maybe later on you will learn it in the other parts of your biology. Okay, so when a cell is lost control, okay, it will actually choose to commit suicide, so that the organism, okay, is not affected. Okay, but sometimes again, a higher degree of control is lost. Okay, and the cell cannot commit suicide. Okay, then the cell becomes cancerous. Okay, and that's when we start having problem. Okay, but otherwise, okay, the activity in the cell is very, very well coordinated and controlled. And how do we do that? Okay, by controlling the enzymes that carries out all this activity. Okay, so we know that all metabolic pathways in cells are regulated by the activity of enzyme. Okay, anything that happens in the cell requires enzyme. Otherwise, very few reactions are spontaneous. That doesn't happen without an active participation by enzymes. Okay, so we need to regulate enzyme. If we can regulate enzyme activity, then we can regulate metabolic activity. Okay, and how do we do that? Okay, these are 5 basic ways to do it. Okay, we'll look at that in a bit more detail later on. So genes that code for enzyme can be turned on and off. Okay, Have you all done protein synthesis, gene expression? Rasa belum. This is chapter 4 kan? Okay, probably in the later chapter you talk about gene expression. Okay, so a protein is produced. Okay, and enzymes are protein. So an enzyme is produced when its gene is expressed. Okay, so protein dalam sel tu is made, okay, using information contained in the gene. Okay, so when we say that a gene is turned on, it means that the cell is actively using information in the gene to produce the protein. Okay, so in this case, for this example, beta-galactosidase. Okay, so beta-galactosidase is produced by the cell. Okay, when the gene that codes for beta-galactosidase, okay, is allowed to express, and the cell use the information in that gene to make beta-galactosidase. Okay, so that is a top level of what you call it, a top level of regulation. Whether do we want to produce the enzyme or do we don't want to produce the enzyme? Okay, if we produce the enzyme, then it's active. If we don't produce the enzyme, that is not there. Okay? So that reaction does not happen. Okay, so beta-galactosidase is a good example. Okay, because this enzyme is only required, okay, when lactose is present. Okay, so it's required for the metabolism of the of lactose. If there's no lactose, then we have no reason to produce beta-galactosidase. So we shut down the production, okay, in order not to waste energy and waste chemical resource. Okay, so as I said, your body is very smart. Your cells are very smart. Okay, kadang-kadang yang bisa sana tu bukan you orang tapi you punya cell tu. Okay, alright. So the other way is to produce the enzyme by produce the enzyme in an inactive form. Okay, so contohnya pepsinogen. In this form is inactive, okay, but under certain condition, okay, it can be activated into pepsin, which is an enzyme that digests protein. Okay, so usually enzymes that digest proteins are dangerous because, as I said, they can chew anything up. Anything that dia nampak protein dia makan saja. Okay, so if they see any protein, they will start digesting it, including your cellular protein. So you would want to be able to have a very strong degree of control over your enzymes that degrade protein. Okay, so what the body does is it produces the enzyme, but in the form of a inactive form that can be activated later by removing a certain part of the enzyme. Okay, again, we'll take a look at an example later on. Okay, so at another level, you can also control enzyme by using inhibitors. Okay, so you can inhibit the enzyme when you want to switch it off, and then remove the inhibition when you want to switch it on. Okay, you can also activate the enzyme, okay, using an allosteric regulator. When you want the enzyme to be on, activate it. When you want to switch it off, remove the activating enzyme. Okay, and there's also a phenomenon called end product inhibition or feedback inhibition, on how one enzyme can control the rate of reaction for the entire pathway. Okay, all right, let's take a look at this one by one. Okay, so again, just to recap, okay, enzymes are powerful molecules, okay, and the activities must be carefully controlled. Imagine your acetylcholinesterase suddenly run out of control. Okay, they refuse to work. Okay, so you would have a paralytic state. Okay, so they need to be controlled to prevent harmful effects and to prevent wasting energy and resources. Okay? And there are basically two major pathways to control enzyme or to regulate enzyme. Okay, so for inducible enzymes, these are enzymes that the cell will only produce when it's required. Okay, the example was the beta-galactosidase. So the cell only produces the enzyme when there's lactose present. Otherwise, no point, because beta-galactosidase only works on lactose. Okay, it breaks down lactose, but without lactose, then there's no need for it to be around. Okay, so you control that at the gene expression level. Okay, bila ada substrate, when the substrate is around, then produce the enzyme. When the substrate is not around, okay, you don't need the enzyme, then do not produce it. Okay, that allows you to control it at a high level. Okay, on the other hand, constitutive enzymes, these are enzymes that are important and they're always around. Okay, they need to be always around. The cell keeps making them. They're always in the cell. Okay, So they are regulated by using activators. Oh, sorry. Wait, where did my PDF go? Okay, sorry, here. Okay, they are regulated by activators or inhibitors, or sometimes via covalent modification. Okay, so reversible inhibition, and also sometimes using reversible covalent modification. Okay, all right, let's take a look. Any questions so far? You okay there? All right, okay. Okay, any questions, just ask. Okay.
Prof,
Yes.
I want to ask for irreversible inhibitor.
Okay.
Okay. So for irreversible inhibitor, they also have competitive and non-competitive?
Usually no, because they don't need to compete. They just destroy the enzyme.
Okay.
So they will act as non-competitive.
So the major for irreversible inhibitor is toxin, right?
Toxin is one class. Okay, but there are also other classes of irreversible inhibitors.
What is the another class?
There are actually several classes. I can't quite recall. Okay, but toxins are not all toxins, but most animal venoms they will contain some of these what you call it inhibitors. Okay, irreversible inhibitors. Okay? And they mainly act on your neural system. Okay, so they are good examples when we are giving lectures. Okay? But bear in mind that there are also many types of enzymes. Okay, I think by now we know about not less than 20,000 types of enzymes. Okay, and there can be many different types. Okay. Okay, thank you for the question.
Thank you.
Okay. Okay, so you start by allogenetic regulation. Okay, so this is where the regulating molecule bind briefly or reversibly to a site other than the active site. Okay, I think we have seen that a moment ago. So and by binding to the site, okay, it changes the shape and function of the active site. Okay, so it can activate or it can inhibit. Okay, so when the regulator binds to the enzyme, it can make the enzyme active, or on the other hand, it can make the enzyme inactive. Okay, in this sense, it becomes a form of non-competitive inhibition. Okay, okay, and this is how it works. Okay, a slide on how it works. So, an enzyme there that has many subunits. Okay, so this is one type of enzyme. Okay, where it has 4 subunits. Okay, and it has 4 active sites. Okay. Now, when the enzyme picks up an activator, okay, so an allosteric activator, okay, remember, does not bind to the active site. It binds to another site. Okay, but by binding to the other site, okay, it either stabilize the enzyme or change its shape so that, okay, it's now easier for the enzyme to bind the substrate. Okay, so this activator. Okay, so in this form, in the active form, the enzyme is active, but the activity may be low. As I bind the more allosteric activator, then the activity of the enzyme increases. Okay. On the other hand, okay, the active form can also change into an inactive form. Okay, by binding an inhibitor again at the allosteric site. Okay, so when an inhibitor binds to the allosteric site, okay, it changes the shape of the enzyme, and it can now no longer bind the substrate. Okay, the active sites, the conformation of the active site has been changed, so it no longer binds the substrate, and it becomes inactive. Okay, so this is one way where the enzymes can be regulated, and sometimes you have multiple effects. Okay, where binding and activator, okay, affect all 4. Well, although it's only binding one allosteric site, but all the units are affected. Okay, so and there are also cases where the more activators that you bind to this complex, then the higher is the degree of activation. Okay, so this is what we talked about a moment ago when you have cooperativity between the enzyme. Okay, so this is a type of allosteric activation. Okay, where the response of the enzyme to the substrate is amplified by binding of a substrate molecule to one active site. Okay, so so the enzyme may have 4 active sites. Okay, the binding of one substrate, okay, cause the other active site to be more active. Okay, so it's either you can either bind a substrate or an activator. Okay, although only one of the subunit, okay, is activated, but the rest of the subunit is also affected. Okay. Okay. So that is how you can regulate enzyme activity using activators and inhibitors. Whether you want them to be active or inactive, or you want them to be active to a certain degree, very active, okay, less active, and so on. You can control the degree of activity, okay, by binding either inhibitors, activators, or even the substrate. Okay. Now we know that enzymes seldom work alone. Okay, so enzymes are usually involved in long metabolic pathway. Okay, so one enzyme may do one reaction, okay, and then another enzyme take over, and you have something called a biochemical pathway. Do we have an example here? Okay, this is one example. Okay, so the first enzyme, okay, may work on the first substrate, okay, and change it to an intermediate. Then the second enzyme picks up the intermediate and change it to another substance and create another intermediate. Okay, and the final product is only produced at the end of a long chain of reaction. Okay, this is something that's more typically happen in the cell. Okay, everything is happening in the pathway, and along the way, some of this intermediate can be shunted off to other reaction. Okay, so as I said, in the cell is a very big, very noisy, okay, very busy place. A lot of pathway like this happens simultaneously. And imagine the cell has a control every one single one of these. Okay, so one of the way to control is by feedback inhibition, where, okay, the final product, okay, somehow they inhibit the first enzyme. Okay, so the first enzyme is involved only in the first step. Okay, so this is not the product of the first enzyme, but it's the product of the pathway. Okay, and it can take one full circle, go back to the first enzyme and inhibit it. Okay, so this is called feedback inhibition. Okay, the inhibition happens when there's enough production of the final product. Okay, so in this example, this is the pathway to produce an amino acid called isoleucine. Okay, so isoleucine is an important amino acid in the cell. Okay, especially bodybuilders, they like to take isoleucine. It allows them to build muscle quickly, to burn energy faster. Okay, and build muscle quickly. Okay, so when the cell has sensed that, okay, it has produced enough isoleucine, okay, then the isoleucine in turn can inhibit the first enzyme in the reaction, okay, and shut down the entire pathway. Okay, again, it makes sense because once you have enough isoleucine, okay, then there's no reason to make any more isoleucine. Okay, so the cell will start making isoleucine when there's not enough isoleucine. Okay, and then the whole process begins. The pathway is initiated. But once you have enough isoleucine, okay, then it's time to shut down the pathway by inhibiting the first enzyme in the process. Okay, all right. So so let's say, okay, I make isoleucine. Okay, intermediate A, intermediate B, intermediate C, intermediate D, and okay, I get isoleucine. Okay, when I have enough isoleucine, okay, I go and inhibit the first enzyme. Okay, I shut down the process. Okay, then tomorrow I find that I need more isoleucine. What happened? Anybody want to try to make a guess? What happened now that you have enough isoleucine? So you can switch it off. When your isoleucine runs out, petang ni pergi gym, burn out a lot of energy, habiskan isoleucine. So tomorrow you need more isoleucine.
So the isoleucine would be released from the enzyme?
Yes, precisely. Thank you. So when the concentration of isoleucine goes down, okay, then it tends to be released from the enzyme. Okay, so now the inhibition is released. Okay, and the pathway can continue again. Okay, so examples of feedback inhibition, okay, happens in many pathways. especially where in a pathway that produce important end products like amino acids. Okay, so the end product itself will inhibit the pathway. Okay, and it makes sense that when you want to inhibit the first enzyme, okay, so that the whole pathway is switched off. Okay. The first enzyme must start the pathway. Without intermediate A, the pathway cannot go on. Okay. So when you cut off the first enzyme, then the rest of the enzymes are also not required. Okay, you don't have to worry too much. You completely switch down the pathway. Okay, so that's feedback inhibition. Okay. Now, covalent modification. Okay, this is not in your notes, so I'm not sure whether it's you need it for your exam or whatever. I just include it because it's part of the thing that we always talk about when we talk about enzyme. Okay, so there are certain chemical modification that you can do to an enzyme, okay, to activate or inactivate it. Okay, and one of the most common example is to phosphorylate an enzyme. Okay, as the name suggests, okay, when you phosphorylate an enzyme, okay, so you what happened is that, okay, a phosphate group is transferred from ATP, usually ATP, but it can be other phosphate carrier, okay, to the enzyme. Okay, and the enzyme now has a phosphate group, a very small phosphate group. Okay, but that reaction, okay, can change an enzyme from an inactive state to an active state. Okay, so again, it's a switch on. Okay, and this is actually very common in the cell. Okay, there are a large group of protein kinases, okay, whose sole function is to phosphorylate enzyme. Okay, so when you add an phosphate group, it becomes active. Okay, when you remove the phosphate group, it becomes inactive. So that gives you a very fine level of control. Okay, a good example is the enzyme glycogen synthetase. Okay, so when there's a lot of sugar around, okay, then glycogen synthetase will be activated by receiving a phosphate group, and it can start to collect the sugar, the glucose, okay, and make them into carbohydrate, okay, starch, glycogen. Okay, when there's not enough sugar glucose around, then the enzyme is dephosphorylated. The phosphate is removed and it's no longer active because there's no more sugar around. Okay, it can also happen the other way. Some enzymes, okay, is opposite. So the inactive form has a phosphate group. Okay, and when that phosphate group is removed by another class of enzyme called phosphatase, then the enzyme become active. Okay, so it can either happen this way or the other way. Okay, phosphorylation can lead to an active enzyme or leads to an inactive enzyme, depending on the enzyme. Okay. So this is actually one of the most common way that enzyme is controlled in your body, and it's very important. Okay. A lot of disease due to cellular metabolism, like cancer, okay, are due to errors in phosphorylation. When the wrong enzyme becomes phosphorylated, or when the enzyme is phosphorylated and not dephosphorylated, and so on. Okay, so that later on when you become doctors, okay, you'll find that, okay, a lot of diseases are actually due to something that happened in your body, something that went wrong with the system in your body. Okay, so this is another form of my modification control. And one more thing we look at are coenzymogens. Okay, so these are the enzymes that are produced in an inactive form. Okay, these are usually very powerful enzymes like trypsin, okay, protease enzyme, like lysozymes. Lysozymes break down unused protein in the cell, so they must be precisely controlled. Okay, otherwise they will break up everything, good or bad protein. Okay, they will just remove. Okay, so chymotrypsin is a digestive enzyme. You use it to digest your food, and they're produced in the pancreas as the inactive form. Yes, because you don't want them to be active in your pancreas. You don't want them to destroy your pancreas, and you end up without pancreas. Okay, all right. So they are produced in inactive form in the form of chymotrypsinogen. Okay, and then they're transferred to your duodenum. Okay, are you all familiar with human biology? You know you have a pancreas, right?
Yeah.
Okay,
Yes.
where's your pancreas? Tak tahu. Where's your pancreas? Lutut.
Near to the stomach.
Near to your stomach. Yeah. Which side, left or right?
Left.
Left. Usually it's on the left. Okay, just above your stomach, under your rib cage. There's a pancreas. Okay, is it important to you?
Yeah.
Yeah.
Okay. What does it do? You don't know. Okay. There's two very important functions. Okay, one is to produce enzyme. Okay, and also the second one is to produce insulin. Okay, you remember. All right. Okay, and then the duodenum. You know where is the duodenum?
Yes.
Okay, this is your part of your usus kecil, your small intestine. Okay, that's where your food digestion takes place. So it's logical that the enzyme is transported to the duodenum. Okay, only then, when it's in the duodenum, when it's ready to carry out its function, then it's converted to the active chymotrypsin by another enzyme, trypsin. Okay, so basically, what trypsin does is it removes two very small portion of the enzyme. Okay, it will cut and remove these two small parts of the enzyme, and then without that, the enzyme becomes active. Okay? It can now start to degrade protein in your duodenum, okay, without causing harm to your pancreas. Okay. All right. Okay. Okay. So I think we have done with regulation of enzyme, inhibition, activation. Okay. Allosteric. Okay. Reversible and non-reversible inhibition. Okay. Covalent modification, feedback inhibition, Zymogen, and so on. Any question on that?
Prof, I have one question.
Okay.
Are all enzymes considered to be allosteric enzyme, or do we have non-allosteric enzyme?
Again, okay, so there are many types of enzymes. So we do have enzymes that are allosteric, okay, and we do have enzymes that are not allosteric. It all depends on whether you want to control the enzyme or not. Okay. For example, like the chymotrypsin that we just talked about, okay. So once it's in the duodenum, okay, there's really no need to control it. You can just let it go and let it be the blasta or the protein sinusoide, because in the end, that's what you want to break down the protein that you eat into amino acid that you can absorb. Okay, so chymotrypsin will be an enzyme that you do not control, okay, using allosteric mechanism. Okay, so it all depends. Okay, so the human cell tend to be a bit more what you call of a control freak. So it insists on controlling a lot of enzyme in the cell. Okay, but some cells, some organisms like bacteria, so they release a lot of enzyme into the environment. Okay, so for that you don't have to control. Like bacteria will release amylase into the environment to break down starch so that it can eat the glucose. Since it's outside the cell, so the bacteria doesn't control it. So those types of enzymes tend not to have any allosteric site. Okay. It just have to be active, do it work, and at the end go away. Okay, all right. Okay, we move on to the next topic. Yes.
Some enzymes are tertiary structure protein, and some enzymes are quaternary structure protein. So is it true when I say that all the tertiary structure enzymes, they are not allosteric enzymes because they are not formed by combinations of subunit protein?
I don't think we can generalize, because even a protein with only one subunit, okay, it can have an allosteric site.
All right.
There are so many enzymes out there. Then so probably there will be some enzymes that exist as one subunit, and then it can be controlled allosterically.
Okay, thank you.
Okay, thank you. Okay. All right, so we quickly go to the next section on classification enzyme. Okay, so as I mentioned in sometime before, okay, so enzymes names are usually has an ASE at the end. Okay, dehydrogenase, oxidase, reductase, and so on. Okay, and based on common classification, okay, we recognize 6 classes of enzyme. Okay, based on the type of reaction that they catalyze. Okay, and this classification is based on recommendation of a body known as the Nomenclature Committee of the International Union on Biochemistry and Molecular Biology. Okay, it was formed in 1964. Okay, and this is the authoritative body that will give a name to the enzyme. Okay, so you don't simply give a name to enzyme. even though you may discover the enzyme. Okay, so every year many enzymes are discovered. Okay, you can propose a name, but the final name will depend on the on this committee. Okay, so they will decide what type of name that is suitable for the new enzyme and so on. Because there's so many enzymes around, and you do not want to have any ambiguity. Okay, I call you Farah, you pun call your enzyme Farah, then banyak pula Farah nanti. Okay, all right, and these are the 6 classes of enzyme, 6 general classes of enzyme. Okay, so the first class is an oxidoreductase. These are enzymes that carry out oxidation reduction, in which oxygen and hydrogen atoms are gained or lost, or transfer of electron. Okay, so the function of this enzyme are mainly in energy transfer. Okay, where they transfer oxygen and hydrogen atoms. Okay, so an example is an NADH dehydrogenase. Okay, the second group of enzymes are known as transferase. Okay, so this group transfer functional groups from one substrate to another. Okay, an example is hexokinase. And we have hydrolases. Okay, so a class of enzyme that catalyze the hydrolysis of water of a substrate. Okay, and an example is dipeptidase. Okay, and then we have lyase. Okay, which form or remove double bonds with group transfer. Okay, an example are fumarase, PEP, carboxylase. And we also have isomerase. Okay, which rearrange the configuration of atom within the molecule. So it converts a molecule from one isomeric form to the other. Okay, and an important example is the triose phosphate isomerase. So, and we have ligase. Okay, these are enzymes that join 2 molecules using energy derived from the breakdown of ATP. Okay, an example is DNA ligase. Okay. So there are many examples, okay, and you don't really have to memorize all of them, okay, but just make sure you know that there are 6 classes, okay, and what are the general features of each class. Okay, so enzymes are not only important to our human body, okay, we have started to make use of them in many applications in industry and medical. Okay, as I think we have one, we mentioned one just now, where proteases are used in the detergent that you use to laundry your clothes. Okay, so kalau you tertumpah kari kat your jeans ke, okay, ada kesan ke, okay, so it's the protease in the detergent that break down the protein and allow you to wash your jeans clean. Okay, so these are examples of application. Okay, for example, in the baking industry, the protease is used to break down protein in flour for biscuit manufacture. Okay, so you need a low protein flour. Okay, so we use a protein to break down the protein in the flour. In the paper industry, okay, we use enzymes called ligninases from fungi to remove lignin from pulverized wood. Okay, so wood is basically made out of 3 components: cellulose, hemicellulose, and lignin. And lignin are very tough molecule to break. Okay, so in fact, very few organisms can break down lignin, and some of these are fungi, kulat. Okay, they produce enzymes called ligninase, and this can break down lignin in your wood so that you can crush the wood to very fine fibers and use them to make paper. Okay? That's why kertas sekarang you buy paper now, you get very nice smooth paper. Okay? Zaman saya paper tu kasar sikit. Okay? Now we have better ligninase to remove the lignin. Okay, and in the food industry, a lot of enzymes are used. Okay, one good example is glucose isomerase. Okay, this is used to manufacture glucose, fructose syrup from glucose. Remember just now class 5 isomerase. So a glucose isomerase is an example of a class of a type 5 enzyme, class 5. Okay, it changes glucose into fructose. Okay, now why do we want to do that? Manufacture of fructose syrup from glucose. Both of them are sugar. So why bother taking one step to change glucose into fructose? Anybody want to make a guess?
Fructose is more sweet and making less amount.
Yes, thank you. Fructose is much more sweeter. Okay, in fact, fructose is about 6 times sweeter. Okay, and manusia ni suka benda yang manis kan. Okay, so you can sell fructose, okay, at a higher price. Okay, in milk production, okay, so we can use lactase, okay, to produce lactose-free milk. Okay, I think that's another example. Somewhere down there, we'll talk about this later on. Okay, and in the medical field, okay, so protein degrading enzymes such as trypsin, okay, that can be used to remove blood clots, okay, and in cleaning your wounds, okay. So blood clots are made from protein, and you can use trypsin to digest them away so that you can clean the your wounds properly. Okay, these are just some examples. Okay, there are many other examples. Okay, and in fact, this is a booming industry. Okay, so if any of you have an entrepreneur mind, you may want to look into, okay, how to use bringing what you call it, enzymes for industrial and or other uses. Okay, banyak duit boleh buat tu. Okay, all right. So we are on our last topic. Okay, on enzyme technology. Okay, so we have seen some examples of how enzyme can be used in industry, medical, and a lot of other what you call aspect of life. Okay. Okay. For example, right in the beginning, we see how enzymes can be used for the diagnosis of COVID infection. Okay. So that's what scientists do. Okay. You find solution, you find application. Okay, and use them to improve the quality of life. Okay, so enzymes can be extracted from cell and used to catalyze a wide range of commercially important processes. We've seen some of them. Okay, and there are constantly new processes being developed to manufacture bulk and high added value products. Okay, so contohnya we are trying to make biodiesel from diesel. Right now, it's still a chemical process, not very clean. Okay, but there are people trying to develop an enzymatic process where we can produce biofuel from palm oil. Okay, so tukar palm oil into a maybe biodiesel that can be used as a bahan api, a fuel for transport. Okay, and one of the recent what you call it, a good innovation, okay, in the enzyme technology is the use of immobilized enzyme. Okay, where enzymes are attached to or retained within insoluble or inert support. So we in the cells the enzyme run free. Okay, some of them are attached to membranes, but in general they float around the cytoplasm. When you extract them, okay, you may want to attach the enzyme to some support so that you can have advantages like you can reuse them later. Okay, so we look at an example later on. Okay, so one of the advantages of immobilized enzyme is that they can be reused. Okay, and continuously produced. Okay, they do not contaminate products, and they're generally more stable when they are immobilized. Okay, the disadvantages are that sometimes when you immobilize an enzyme, you can have a loss of activity because some immobilization technique requires you to bind the enzyme to a support, and that can affect the activity of the enzyme. Okay. All right, and you can also change the kinetic of the enzyme. Okay, between a free-floating enzyme and an enzyme that's immobilized on some substrate. Okay, so the activity of the enzyme, the kinetics might change a bit. The Vmax and the Km might change a bit. Okay. Okay, and this is some methods to immobilize enzyme. By immobilizing enzyme, we mean that the enzyme is no longer freely soluble in a liquid. Okay. So you attach it to some solid substrate. Okay, so you can either do by absorption. Okay, so for support, this can be some plastic resin or so on. Okay, and the enzyme will just naturally adsorb to it. Okay, by natural hydrogen bonding and so on. Okay, so when you have forces, or any interaction. Okay. So you can use things like ion acceleration and hydrophobic resin. Or you can entrap the enzyme in a three-dimensional lattice. Okay, the enzymes are not chemically bound, but they are trapped between you can imagine a sponge, something that has a lot of holes. The enzyme can be trapped between that. Okay, and you can use things like polymers, alginate and silica gel. Okay, we'll discuss an example using alginate in a short while. So the enzymes are trapped between something, but they are not chemically modified. In these two cases, the enzymes are not chemically modified. They either just stick to something, or they're trapped in some kind of matrix. Okay. So this is one example of using immobilized enzyme. Okay. So milk is a good food. okay, but it contains lactose as a sugar instead of glucose, okay. And some people has intolerance towards lactose, okay. For example, I have a slight a mild case of lactose intolerance. I cannot drink too much milk, okay. Otherwise, I have to move to the toilet a lot of time, okay. So one way for people like me to enjoy milk is to consume lactose-free milk, okay, where the lactose is removed by using the enzyme lactase. Okay. So what do you think lactase do? Tengok nama boleh suggest tak? Lactase. What does it do?
Break down lactose.
Sorry?
Break down lactose.
It break down lactose into suara
Small
bertubi-tubi ni.
glucose.
Glucose and galactose.
Glucose and galactose. Okay, very good. Okay. Okay, so lactase will break down lactose to glucose and galactose. Okay, and that solves the problem of having lactose intolerance. So one way of doing it, you can just add lactase to the milk. Okay. Yeah, so just buy the milk, then buy lactase and add a spoonful of lactase to the milk. Wait for the enzyme to work. By then, the lactase will be in your milk. Okay, and you'll be drinking lactase with your milk. Okay, and the lactase itself may cause an allergic reaction. Okay, so tak ada lactose, okay, tapi allergic ke lactase pula. Okay, okay, same story. All right. So you don't want that to happen. Okay, and then also you waste the lactase. Okay. So the other way to do it is to immobilize the lactase on a solid support. Okay, so you can look at this diagram. All right, so this is you can imagine this is a glass support. Okay, with a lot of resins. Okay, you can imagine small bits. Okay, on each small bits on the surface of each small bits are full of lactase. Okay, they are covalently attached to the bits. Okay, so the lactase remain on the bits. Okay, they are attached to the bits, and then you can just pass your milk through it. So as the milk goes through it, okay, the lactase on the bits will start degrading the lactose into glucose and galactose. Okay, and then it comes up from at the bottom into and make your lactose-free milk. Okay, but your lactase will not come out because they are immobilized on your resin. Okay, they remain here lactase. Okay, so that, okay, you can use it over and over again. Okay, you don't waste the enzyme, and also the enzyme does not end up in your product. Okay, you can sell your lactose-free milk, okay, without any trace of lactase. So this is an interesting practical. Kalau boleh you pujuk your lecturer, okay, buat dalam makmal. Okay, you only need some lactase and some calcium arginate. Calcium arginate ni macam is like agar-agar. You boil, okay, you can dissolve it in water. Okay, so it becomes solid. Okay, the moment you add some calcium. Okay, so calcium arginate, you can dissolve it in water. It looks like a water, okay, liquid. But when you add calcium, then it begins to solidify. Okay, so it's an interesting experiment. Yeah. So you can mix it. You can add arginate to water, dissolve it, and then you add your lactase to the same solution. Okay, and then you just take it up and drop it into a beaker of calcium chloride. Okay, and you will immediately solidify into small bits, like what you see here. Okay, so this is an example of the use of immobilized enzyme, where enzymes are immobilized onto a solid support. Okay, and okay, so besides absorption and entrapment, there are also other ways to immobilize the enzyme. Okay, these are ways that might modify the enzyme, make changes to the enzyme. For example, you can bind the enzyme to the resin or support by using covalent bonding. Okay, so this is where you form a chemical bond between the enzyme and the support material. Okay, very strong bond, so the enzyme will not easily detach. Okay, but as mentioned, the disadvantage is that it might change the enzyme a bit. Okay, because now it's chemically modified. Okay, or you can do cross-linking, where you can covalently join the enzyme to each other, and they can form a small grid. Okay, so examples of support that you can use is like glucoaldehyde. Okay, or the more popular way now is by encapsulation, where you encapsulate or cover or wrap your enzymes in a semi-permeable membrane. Okay, for example, nylon or cellulose nitrate. Okay, so this is such that the capsule, the pills that you take, there's an outer layer, and inside is the enzyme. Okay, and your substrate will diffuse through the membrane. Okay, and the product will diffuse out. Okay, so I think this is a slide to summarize all this method of how you can immobilize your enzymes. Okay, by either absorption, okay, entrapment, where the enzyme is not modified. Okay, or you can cross-link the enzymes together, or covalently bind it to a support. Okay, this is where your enzyme might be chemically modified and lose some activity, or you can encapsulate it within a semi-permeable membrane. Okay. Okay, and I think with that we are done for this topic. So, is there any question or is there any clarification that you require? I'll be happy to answer.
Prof, may I ask a question?
Yes, sure, please.
The part where the disadvantages of the immobilized enzyme, where it changes the kinetics of the enzyme, is that true to say that when the kinetic of the enzyme is changed, the rate of collision between the enzyme and the substrates will be decreased and less product will be formed?
That is one way of saying it. Yeah, so what can happen is that when you covalently bind the enzyme to the support, okay, so the enzyme is no longer free to move. So the active site might be partially covered, or the allosteric site might be partially covered. Okay, and you lose some activity because of that. Okay. On the other hand, it is also possible that covalent binding can actually increase the activity of an enzyme. Okay, so these are the things that we need to experiment on. Okay, sometimes you cannot predict which direction it will go to. Okay. But usually, when you modify an enzyme by covalent bonding, you tend to lose some activity.
Thank you, Prof.
Welcome.
Okay, so anyone has more question for Prof? Okay, Prof, I do have one question. So this question is actually from our YouTube channel, from our student. So the question is, based on the enzyme used in the swab test, can enzymes be manually produced?
Definitely. Sorry, is there any further part to the question?
No, that's all. Yeah,
okay. Definitely, those enzymes are produced. Okay, so there are two ways of doing that. You can either both enzymes are produced in bacteria. Okay, so especially the DNA polymerase comes from a bacteria called Thermus aquaticus. So it's a bacteria that we isolated from hot spring. Somebody isolated from hot spring. That's why it lives in a very hot environment. Okay, that's why it has such a high optimum temperature. So you can either culture the bacteria, the Thermus aquaticus, okay, in large volume and get it to produce the enzyme. Or more commonly now, we use genetic cloning. So we clone the gene. Okay, we clone the gene from the bacteria. We know after a lot of research, we know that which gene codes for this protein. We can actually take the gene out from the Thermus aquaticus, okay, and insert it into a simpler bacteria or something that is easier to work with. For example, Escherichia coli, E. coli, okay, and get E. coli to produce the enzyme. Okay. So both the enzyme now, the reverse transcriptase and E. coli, and also a lot of enzyme that we use are actually produced by gene cloning into other organisms. So we definitely have the ability to produce enzyme on a large scale.
All right. Okay. Thank you so much, Prof, for that answer. So anyone else? So maybe you all have any question for Prof before we end our session today?
Prof, I have a question.
Go ahead.
Is it possible for us to modify an enzyme so that maybe it can withstand a higher temperature?
Okay, thank you. That's a good question. And the answer is again, definitely. Okay, in fact, again, that is a very active field in biotechnology now, enzyme engineering. Not only you can change the enzyme to make it work harder, higher rate of reaction, you can also change that enzyme so that you can expand the type of substrate that you can take. Okay, you can modify its product. Okay, there's a lot of things that you can do the enzyme now. Yeah, with our current technology in gene manipulation and protein engineering. Okay, so in fact, that's what the SARS virus, the COVID-19 virus did. Okay, it changed one of its protein, and because of that, it is able to infect human. Okay, so by genetic mutation, okay, you can change enzyme. Okay, whether in the test tube in the lab, or you can do it in the organism in the lab. Okay, and come up with an entirely new enzyme. You can even combine several enzymes into one enzyme. Okay. So the technology has gone very far. It's a bit scary. Yeah, there's a lot of ethical concern about it, but all these are possible now, okay, and very exciting.
Yeah, that's very interesting. Okay, all right. So, anyone has more question?
There is two more questions in YouTube. First, what is the difference between allosteric inhibitors and irreversible or reversible inhibitors?
Okay, so an allosteric inhibitor is an example of a reversible inhibitor. Okay. So, but it binds to the allosteric site. Yeah. So another example of a reversible inhibitor is the one that binds to the active site. So that's probably the simplest way to look at it.
Yeah, so the one more question is: Are they ways to overcome irreversible inhibitors?
Sorry, can't get you.
Are there any ways to overcome irreversible inhibitors?
In general, irreversible inhibitors, as the name suggests, so you cannot reverse it. Okay, although in some cases, when somebody is poisoned by an irreversible inhibitor, okay, so the doctor will attempt to give him some other chemicals that can break the bond. Okay, but that is usually not the suggested way, because it might cause a reaction that we do not know. Okay, so usually when you are, for example, you are poisoned by an irreversible inhibitor, what the doctor will try to do is actually to provide the enzyme to you. Okay, so whether the enzyme, your body can actually fight if it has the time to produce more enzyme. Okay? So now your enzyme is irreversibly inhibited, so you can't use it. Okay, if your body has a time to produce more enzyme, okay, that will save you from poisoning. Okay, so in general, if you ask me the risk of what you call it, removing an irreversible inhibitor, that's a bit difficult to answer. But I would say that there will be some risk, okay, because the chemical is covalently bound to the enzyme, so breaking that bond may cause some damage to the enzyme.
All right.
Okay, thank you very much, Prof. All right, so I think that's all the questions for today. I think that's all the questions that we have for today. So thank you so much, Professor Faiz, for such a brilliant and extensive explanation about enzyme. And as Prof has said, enzymes are very powerful molecules that have many purpose in biotechnology, and it's just not a mere molecules that work in our body. So I think this is the end of our lecture today, and I hope this session is helpful and beneficial to all of us, and I believe everyone is learning a lot from Professor Faiz today. Again, thank you so much, Prof, for spending your time with us, and I do hope we can have you again sometimes in the future, Prof.
Inshallah.
Would you come again?
Thank you very much for joining me. Okay, and I hope that you learned something. Okay? And all the best in your future endeavors. Okay.
So okay, we learned so many. Yeah, Prof.
Yes.
Yeah, Prof.
Just one final advice. So when you learn, okay, try to expand your horizon. Okay, think of what other things that can happen. Okay, and not really what is in the lecture notes. Okay? So that's where the real learning happens.
Yes.
Okay, so
yes.
thank you very much for inviting me.
Thank you, Prof. Yes, that's very true, Prof. All right. So thank you, Prof, and thank you all lecturers and all students for joining us today. And for students, you can find your attendance link in the chat box, and also the QR code will be displayed also on the screen. All right. So thank you so much, everyone. Stay safe. And we're going to be dismissed. So thank you and goodbye and assalamualaikum.
Waalaikumsalam. Okay, thank you, everyone.